Welcome back to The Explainer. If you're grinding through prep for your NCLEX, MCAT, or USMLE, you know how crucial it is to nail down the basics. And today, we're cutting right through the noise. We're going to look at exactly how a perfectly normal, healthy cell goes rogue and transforms into a cancer cell. It's an absolutely foundational concept, and we're going to break this journey down step by step with some crystal clear visual frameworks to make sure it actually sticks in your brain.
1 trillion. Just let that staggering number sink in for a second. Every single day, your body is churning out about 1 trillion new cells. And, with that massive amount of division, it's basically a statistical guarantee that some of those cells are going to make a mistake when copying their DNA. Now, usually, no big deal. Your body catches the error, eliminates the bad cell, and we move on. But sometimes, it doesn't. And that one missed mistake, multiplying silently under the radar for years and years, well, that is the literal genesis of cancer. So here's our roadmap for today. We'll start with cellular adaptation, move into the genetic tipping point, then cover the hallmarks of cancer, classifying tumors, and finally, we'll wrap it all up with a clinical tie-in. All right, let's dive right into part 1, cellular adaptation, the normal response. See, before we can even begin talking about cancer, we really have to understand our baseline. How do our cells normally adapt to stress? Pathologists look for 5 key adaptive responses, and you absolutely need to know these cold for your exams. The first step is hypertrophy. Now, this is strictly an increase in cell size. The cell is not dividing here. It's simply getting larger, kind of beefing up to handle a heavier workload. It's exactly like a bodybuilder's muscle cells getting massive with heavy resistance training. Clinically speaking, a super common example you'll see is the left ventricle of the heart thickening up. It does this in response to the massive workload of chronic hypertension. Now, you've got to contrast that directly with our second adaptation, which is hyperplasia. This one is an increase in the actual quantity, the sheer number of cells. And this can only happen in tissues where the cells actually still have the ability to divide. Think about the endometrium thickening up every month under the stimulation of estrogen, or say getting a tough callus on your hand from the repeated friction of lifting a barbell. So remember, hypertrophy is all about size, but hyperplasia, that's about quantity. Third on our list is atrophy. You can think of this as the body basically downsizing to save on resources. It's a decrease in either cell size or cell number, and it happens because of disuse, a poor blood supply, or maybe nutrient deprivation. The most striking visual example of this is the muscle wasting you'd see in a patient's limb right after a cast gets taken off after 6 weeks. If you don't use it, or if it's starved of nutrients, the tissue just atrophies. Okay, next is metaplasia. And this one is just a fascinating survival mechanism. It happens when one fully differentiated cell type is completely swapped out and replaced by a totally different cell type. Why? Because the new one is better equipped to handle a specific ongoing stress. The classic, highly tested example here is a chronic smoker's airway. The delicate, normal ciliated columnar epithelium of the bronchi, it gets completely replaced by this tough, rugged, squamous epithelium. Now, it's a great protective move in the short term, but as we're going to see, it comes at a pretty dangerous long-term cost. And that cost often leads us right into the 5th adaptation, dysplasia. This is disordered cell growth. If you look at this under a microscope, these cells vary wildly in size, shape, and organization. It's a mess. This is the exact moment where microscopic changes become really concerning, because dysplasia is fundamentally a precancerous change. But, and this is vital to remember for your boards, at this stage, it's still reversible if you remove the stressor, like if they stop smoking. The cells haven't quite crossed that point of no return yet. This actually creates a very specific sequence you need to memorize. We start with normal tissue, move to metaplasia, and then into dysplasia. With each step, things are getting progressively less controlled. Now, once you progress past dysplasia into carcinoma in situ, the cells are frankly malignant, but they are in situ, which literally means they're still in their original place. They haven't broken through the basement membrane. The definitive crossing into invasive cancer happens at the exact second they break through that basement membrane, because that's when they gain access to the blood vessels and lymphatics. Moving right along to section 2, the genetic tipping point. What goes wrong? So, what actually pushes a cell over the edge from reversible dysplasia into irreversible invasive cancer? Well, it fundamentally comes down to damage in 2 opposing categories of genes. I want you to think about this like you're driving a car. On one side, we have proto-oncogenes. Normally, these genes just promote healthy cell division, but when they mutate, they become oncogenes. Think of them like a gas pedal that's just jammed to the floorboard, constantly demanding the cell to divide and divide. HER2 and breast cancer and RAS and solid tumors are your classic examples here. On the flip side, we've got tumor suppressor genes. These are your car's brakes. Their job is to stop the cell cycle the moment they detect DNA damage. But when they mutate, your brakes are completely broken. And hands down, the most important set of brakes in your entire body is the p53 gene. It is so absolutely critical that scientists famously nicknamed it the guardian of the genome. Normally, if a cell is just too damaged to be repaired, p53 steps in and triggers apoptosis, programmed cell suicide. But here's the terrifying part. p53 is mutated in over half of all human cancers. When the guardian is knocked out, these highly damaged, super dangerous cells that really should die are just allowed to keep right on dividing. Generally speaking, for cancer to really take off, it requires both things, that stuck gas pedal and the broken brakes. Now, for tumor suppressor genes, you'll often hear about the two-hit hypothesis. This basically means you typically need both copies of the brake gene to be knocked out before you completely lose its function. Oh, and also keep in mind your DNA repair genes, like BRCA1 and BRCA2. Now, they don't control cell growth directly. Their job is to fix the DNA itself. So when a BRCA gene is mutated, genetic damage starts accumulating at a terrifying rate, wildly accelerating that whole pathway to cancer.
Which brings us to Section 3, the hallmarks of cancer.
So how does all this microscopic damage build into the broader, real-world behavior of a tumor? Well, using the legendary framework developed by researchers Hanahan and Weinberg, we can see that cancer isn't just one single thing going wrong. It's more like a cell acquiring a terrifying set of superpowers. Let's systematically break these down. First, self-sufficiency in growth, meaning it drives its own proliferation without even needing any external signals. Second, insensitivity to anti-growth signals. It just completely ignores the body screaming at it to stop. Third, evading apoptosis. Thanks in large part to those p53 mutations, it dodges cell death entirely. Fourth, limitless replicative potential. Normal cells eventually hit a division limit, right? But cancer cells reactivate this enzyme called telomerase to constantly rebuild their telomeres, letting them literally divide forever. Fifth, sustained angiogenesis. It forces the body to build brand new blood vessels just to feed the tumor. And finally, tissue invasion and metastasis. The big takeaway here? Cancer represents a cell that is systematically breaking every single one of the body's normal control systems, all at the exact same time.
Okay, let's look at Section 4, classifying tumors, grading and staging. Once those control systems are broken and a tumor actually forms, we have to rely on some heavy-hitting clinical distinctions. These are the tools pathologists and oncologists use every single day. First up, we've got to draw a really sharp line between benign and malignant. Benign tumors, that is the good news on a pathology report. They grow slowly, they're well differentiated, which means they still look and act a whole lot like their original tissue, they stay wrapped up in a capsule, and most importantly, they do not spread. Think of something like a simple uterine fibroid. Malignant tumors, on the other hand, they grow rapidly. They're poorly differentiated, meaning they look like chaotic alien cells under the scope. They don't have a capsule, they invade local tissue, and they metastasize. So the really crucial point here is how we actually measure that malignancy. And listen up, because this is exactly where test writers will try to trap you. Grading and staging are completely different axes. They are not the same thing. Grading is strictly about what the cells look like under the microscope. It's a measure of differentiation, scaling from a well-behaved grade 1 all the way up to a highly aggressive, completely undifferentiated grade 4. Staging, however, is anatomic. It's a measure of how far the cancer has physically spread inside the body, usually using the TNM system, T for tumor size, N for lymph node involvement, and M for distant metastasis. You could easily have a high-grade, highly chaotic tumor that's caught at a very early stage, or a low-grade tumor caught at a very late stage. Do not mix those two up on test day.
Finally, Section 5, clinical tie-in, Barrett's esophagus.
Let's prove your mastery by applying everything we've just learned to a highly testable clinical scenario. Imagine a patient comes in with long-standing, untreated GERD, chronic acid reflux. That acid is a massive stressor. Step 1, the acid constantly burns the lower esophagus. Step 2, just to survive, the normal squamous lining adopts. It replaces itself with tougher columnar cells. That right there is metaplasia, specifically what we call Barrett's esophagus. Step 3, if that acid exposure just keeps going, the metaplasia becomes disordered and precancerous. Boom, there is our dysplasia. And finally, step 4, if left totally unchecked, those cells break through the basement membrane, resulting in full-blown esophageal adenocarcinoma. You just watched the entire cellular adaptation pathway play out right before your eyes in a perfect textbook clinical vignette.
And that brings us to the end of our journey today. You now know the 5 cellular adaptations, the exact sequence to invasive cancer, the deadly dance between oncogenes and tumor suppressors, the core hallmarks of a cancer cell, and exactly how we classify tumors clinically. In our next explainer, we're going to pivot over into infection. We'll be specifically exploring sepsis, how the body responds when a pathogen breaks through, and why it's still such a leading cause of mortality. Make sure you're subscribed for that one. But until then, I want to leave you with this thought. If our cells really require multiple genetic hits to become cancerous, what silent, everyday stressors might be secretly pushing your cells closer to their tipping point right now? Think about it. Keep studying hard, and I will see you in the next explainer.
Don't forget to follow us for more content like this.